Hi children, good morning. My name is Girish. I teach biological sciences for you. Okay, right. So we have nearly 11 lessons in our textbook, Telangana textbook. We have 11 lessons in biological sciences. Okay, let us see the first one. Here see, the first lesson is cells, its structure and function. First lesson is cells, its structure and function. Second one is plant tissues. Third one is animal tissues. Fourth one is plasma membrane. Fifth one is diversity in living organisms. Sixth one is sense organs. Seventh one is animal behavior. Eighth one is challenges in improving agricultural products. Ninth one is adaptations in different ecosystems. 10th one is soil pollution, and 11th one is biogeochemical cycles. Right. So we have 11 chapters in this 9th class biological sciences textbook. Right. The first chapter is cell, its structure and function. Already we have learnt in our 8th class. What is cell, children? Cell is the basic structural and functional unit of an organism. What is cell? Cell is the basic structural and functional. Cell is the basic structural and functional unit. Okay, cell is the basic structural and functional unit of an organism. In the previous class, in the 8th class, you have learnt, and you have already also learnt about the discovery of cell. Okay, like Robert Hooke discovered the cell, Robert Brown, nucleus, yes or no, and cell theory. All these things you have learned. And next one, you have already also learned size and shape of the cells. Size and shape. So are all the sizes are similar, children? No. Or what about the shapes? Shapes are also not similar. Okay, and we have learned about unicellular and multicellular organisms. Unicellular, so uni means single, so the organisms with single cell are called unicellular organisms. And you have already learnt about the multicellular means many cells, multi means many cells. So the organisms with single cell are called as unicellular. Best example is amoeba. And here you can write anything: human beings, cow, goat, rat, buffalo, plants. All these are multicellular organisms. So you have learnt about the cell, its shape, structure, discovery, all these things. Now let us recall some of the activities related to these aspects. For example, you have seen that onion cells. Yes or no? Onion cells are nearly rectangular in shape, while that of cheek cells are circular in shape. So add other examples as the one given above. So you can add one more example like blood cells. So blood cells. So what is the shape of blood cells, children? Round or circular in shape. The blood cells are round or circular in shape in animals and in human beings, right? Second one, muscle cells. Muscle cells in human beings. Muscle cells are spindle in shape. And you can go for the root hairs. Root hairs in plants. The shape of the root hairs is once again rectangular. Like this, some cells are so columnar in shape, cuboidal in shape, like the different shapes are there. Yes or no? Polyhedral in shape, circular shape is given blood cells, spindle shape is given muscle cells, and rectangular shape is root hair cells. Primarily, cells are studied under microscope. We can't see the cell and its structure normally with our naked eye. Yes or no, children? Can we see the cell in our body? Can we see the cell in our body? No. We can see the parts only, but we can't see with our naked eye. We can't see the cell. Then with the help of an instrument called as microscope only we can see. Either it may be a plant cell, a cell, or animal cell, or human being cell. Okay, we can see even unicellular organisms also. We have to use microscope. Already we have discussed this microscope, different types of microscope, like a simple microscope, compound microscope, electron microscope, all these things. So when we observe the cell under a compound microscope, we can see some parts like cell wall, cytoplasm, nucleus, chloroplast and mitochondria. However, when the same cells are observed under the electron microscope, a few other structures become visible. So this paragraph is telling that the difference between compound microscope and the electron microscope. So if you will see the cell under the compound microscope, only some parts are visible. If you see the structure, cell structure under the electron microscope, why? Because the electron microscope is magnification, the resolution is very high. Okay, resolution is nothing but the thing which we can see that will be big, no, that is called as resolution. Yes or no? So the resolution is very high. That is why even the small things, small particles also we can see with the help of electron microscope. If you see under compound microscope, only some parts like cytoplasm, cell wall, nucleus, chloroplast, mitochondria. Anyway, we will learn about these things later. Okay, but if you want to know more detail of these parts, more depth of this of other parts, and what are the other parts that are there in the cell, you have to go for another microscope, that is electron microscope. Okay, to study various cells, scientists have been trying to observe the cells from different parts of plants and animals. So scientists are observing to study various parts, to study various types of cells. So scientists are going for different parts of plants and animals, and they draw their structure, yes or no, and take a photograph of them and make models of them. They have given valuable information. Okay, these have given valuable information about plant and animal cells. So these scientists, due to their work and due to their contribution, okay, they made so many efforts to study the structure of these plants and animal cells. Yes, these have given a valuable information about plants and animal cells. Now here we will study about the model, okay, diagram of the cell. So we will study two types of cells, plant cell and animal cell. Plant cell and animal cell. Okay, children, now see the two diagrams which are on the board represents the two types of cells. The left hand side, this diagram represents the plant cell, and the right hand side, this diagram represents the animal cell. They see 90% of the parts are common, there are slight differences, that's it. So all the organelles shown a typical plant or animal cell will not exist in every cell. For example, chloroplasts are always shown in a typical plant cell, yet all the plants do not have chloroplasts. So what here telling it is, here if you see, in a plant cell the chloroplast is there. That doesn't mean that all the plant cells will have chloroplast. Okay, so what here the statement is, the organelles which are present in the typical plant cell or animal cell, okay, need not to be present in all the plant cell and animal cells. Right? So here they are mostly found in the, if you observe the chloroplast, they are mostly found in the cells of green leaves, tender stems, etc. The organelles that feature in most of the cells are included in this model. The typical plant cell provide a way to study the cells. Once we arrive at such a model, we can compare any cell with it. So the typical plant cell and animal cell means all the parts. So not only a single cell is present in the plant, so many cells are present. In that so many cells, some parts may be missing. For example, if you take the chloroplast is present in the green leaves, tender stems, etc. The remaining parts, the chloroplast is absent. Okay, so the parts, all the parts that are there in this diagram means, so these parts are the parts of the plant cells. These parts are the parts of the animal cell. Now here, what are the comparison? What is the comparison between plant cell and animal cell? There are some questions. What common features do you see in both the cells? What common features? Yes, children, plasma membrane. Plasma membrane is the common one. Mitochondria is the common one. Cytoplasm, the liquid fluid, that is the common one. Golgi apparatus is the common one. Nucleus is the common one. Mitochondria is the common one. So these are the common. But there are some cell organelles which are present exclusively in plant cell. Those are absent in animal cell, like chloroplast. Centrioles are present in animal cell. These centrioles are absent in plant cell. Okay, right. So here, which cell organelles are found exclusively in plant cell? So vacuoles. Okay, so if you'll observe, this is a vacuole. The vacuole is not there. Even if the vacuole is present, the vacuole may be very small. So vacuoles and chloroplast. The answer is vacuoles and chloroplast. Chloroplast is a type of plastid, children, which is present only in plant cell, that is absent in animal cell. Compare the parts of plant and animal cell. Note down the differences. So here, let us go for the difference. The first difference is in plant cell, cell wall. That is the main difference. Cell wall. So if you observe, this is the cell wall, children. The extra layer. Yes, this extra layer will be present here. That extra layer is absent in the animal cell. So cell wall is present. Cell wall is present in plant cell. Cell wall is absent in animal cell. That is the first difference. Let us go for the second difference. The second difference is, in plant cell, plastids like chloroplast are present. Those are absent in animal cell. And the third difference is, what is the third difference? Centrioles are present in, actually that is not. That centrioles are present in animal cell. These are absent in plant cell. So these are the main differences between plant cell and animal cell. Now let us go for the first one, that is cell membrane or plasma membrane. So we have the diagrams of plant cell and animal cell. Yes or no? So that is a cell membrane or plasma membrane. So the plasma membrane is there. The other name for plasma membrane is cell membrane. Other name for plasma membrane is cell membrane. Other name for plasma membrane is cell membrane. Here also the other name is cell membrane. So see, children, this cell membrane is the outer layer. This cell membrane is the outer layer. Here also the cell membrane is present. See, this is the cell membrane. Okay, this is the cell membrane. And here, see, this is the cell membrane. Yes or no? So this cell membrane is common for both. The cell membrane is common for both. But what makes the difference here? Extra layer cell wall is present. That cell wall is absent there. Okay. So in the earlier classes, you have learnt about the cell membrane is the covering of the animal cell. This cell membrane is also present in plant cell. That's another name for cell membrane is plasma membrane. In plant cell there is another layer, just now I told you, that is called as cell wall. So what is the common layer that is present in both the plant cell and animal cell? Yes, cell membrane. What is the extra layer that is present in the plant cell? Cell wall. What is the extra layer? Cell wall. This is the cell wall. Okay, this is the cell wall. This is the extra layer. Now let us go for an activity. Observing the cell membrane. Observing the cell membrane. Take a rheoleaf. So rheoleaf is a type of leaf. Tear it in a single stroke. Tear it with a single stroke. Okay, take a rheoleaf and just tear it with a single stroke. Observe it against the light. You just observe it against the light. Take a small piece of the leaf peel. Okay, so what you have to do? Take a rheoleaf, tear it with a single stroke, observe it under the light. Take a small piece of the leaf peel and observe under the, observe the transparent light colored portion under the microscope. How do you observe? Already in 8th class we have learnt observation of the onion cells, cheek cells. Yes or no? So for that you have to put it on the slide. So take a leaf peel. Okay, this is slide, children. Put it on the slide, okay, and cover it with cover slip. The slide will be like this, okay. Take this and cover it with a cover slip. The cover slip is a transparent portion, and you just observe under the microscope. If you will observe under the microscope, okay, you can observe the membrane. You can observe the membrane. Here, see, you can observe the membrane. So cover it with the cover slip and observe the light portion under the microscope. Draw the diagram what you have seen. So this is the thing we can see when you observe under the microscope. And now put 1-2 drops of here see salt solution on the membrane and leave it for 5 to 10 minutes. So this is the membrane now, and just 1 to 2 drops of salt solution. Children, how do you prepare salt solution? Salt plus water. Yes or no? So for preparing salt solution, take the measurement of 50 ml of water and dissolve 1 or 2 spoons of salt and stir it. So salt solution can be prepared very easily. Take 50 ml of water and 1 spoon of salt and stir it. The salt solution we will get. So once again I will tell here, take a real leaf and stir it with a single stroke. Observe it against the light. Take a small portion of the leaf peel with a light color. So when you stir the leaf, there will be one attachment between the two parts. Okay, that part will be the light colored portion. Now observe under the microscope and draw what did you observe. And second, the next step, and how you can take, you just take the peel and put it in the center of the slide, okay, and drop some water, put a drop of water on it, cover it with a cover slip, and you have to observe. Now put 1 to 2 drops of salt solution and on the membrane and leave it for 5 to 10 minutes and observe. So here see, you can observe real leaf peel cells with membrane. So that is the first diagram, 3A and 3B. If you will put the salt solution, okay, the sunken cytoplasm, plasma membrane and cell wall, clearly you can observe. To observe the cell membrane only we are doing this activity. So once again I repeat, take a real leaf, tear it with a single stroke, observe the light colored portion. How did you observe? By taking a small portion of the leaf and putting in the center of the slide and put a drop of water, cover it with a cover slip and observe. That is the thing. And next one, put 1 to 2 drops of salt solution, okay, and again observe. So you will observe the parts which are there in 3B, plasma membrane, cell wall, shrunken cytoplasm. As this is a plant cell, real leaf means plant cell wall also can be observed, okay. So observe it under the microscope, draw your observation in your notebook, compare the observation of both 3A and 3B and note down the difference. So the 3A is there and 3B is there. So okay, in 3A we haven't seen any shrunken cytoplasm cell wall. A clear picture is obtained. We can see the cell wall here, we can see the plasma membrane here, shrunken cytoplasm if you will add salt solution. This makes the difference. Okay, right. So what is the reason for observing the shrunken cytoplasm? When we put salt solution over the leaf of real water present inside the leaf comes out. That makes the difference. So when you will put salt solution, okay, on the real leaf, the water present inside the leaf comes out. If water present in the leaves comes out, the cytoplasm will be shrinking. Okay, that's right, cytoplasm will be shrinking. It results in the shrinking of cytoplasm along with cell membrane. So the outer boundary of the colored area is actually the cell membrane or plasma membrane, which becomes separated from the cell wall. So the outer boundary is there now. So in this there are two diagrams. One diagram, just we are keeping the water and we are observing, and the other diagram we have kept, okay, salt solution and are observing. When we pour salt solution, 1 or 2 drops of salt solution on this membrane, the cytoplasm will be shrinking. Why the cytoplasm will be shrinking? Why? Because when you keep the salt solution, the water comes out. When the water comes out, automatically the cytoplasm becomes contracted, and that you can observe there. However, we can observe the structure of cell membrane. Only through an electron microscope. So this is very important point. Okay, we can observe the structure of cell membrane only through electron microscope. Okay, right. So now let's go for cell membrane properties. Okay, cell membrane is flexible. Okay, I'm writing here. Okay, cell membrane is flexible. Flexible means yes, it can bend. Okay, cell membrane is flexible. Okay, and you can observe through electron microscope. It is flexible and is made up of mainly lipids and proteins. It is flexible and made up of lipids and proteins. See children, I am telling about the cell membrane only, not cell wall. Cell wall is the extra layer that is present in, yes, where children? That is present in plant cell only. Cell membrane, or otherwise called as plasma membrane, is present both in plant cell and animal cell. Now we have done an activity by taking the rheolith and tearing with a single stroke and observing the light colored portion without salt and observing the portion with salt. We have seen the shrunken cytoplasm. Now let us go further, and this cell membrane can be seen only through electron microscope. Yes or no? Right. So the property, the cell membrane is flexible. That means it can bend, and it is made up of lipids and proteins. It is made up of lipids and proteins. And one more thing, the cell membrane is the outermost layer of the cell that separates cytoplasm from external environment. So this is the cytoplasm, which is so this is, you can see light yellow in color. This is cytoplasm, and this is here also, this is the cytoplasm. So whatever the cell you take, either the plant cell or the animal cell, so this portion which is shaded, the maximum amount of the portion is called as cytoplasm. So this cell membrane, yes, see, this cell membrane separates the cytoplasm from the external environment. This cell membrane separates, see this cell membrane, the line, yes or no, the line separates the cytoplasm from external environment. Okay, this is also known as plasma membrane. Already I told you. The cell membrane defines the shape and size of the cell. Yes or no? Yes, children. The cell membrane, if here the cell membrane is spherical or round, so it determines the shape of the cell and size of the cell. Here also this is somewhat polyhedral, or okay, it determines the shape and size of the cell. So what are the properties of characteristics of cell membrane? One is cell membrane we can observe through electron microscope. It is made up of lipids and proteins. Cell membrane separates the cytoplasm from external environment. Okay, and next one, this cell membrane is very important. Why? Because it defines the shape and size of the cell and protects the cytoplasm from external environment. The internal environment of the cell is different from outside. So the internal environment, you can see it is different from outside. Inside a cell, one finds a very specific composition of various substances which maintains a balance. The cell membrane plays a crucial role. Inside a cell, so many things are there. Okay, so one more important function, this cell membrane plays a crucial role in maintaining the balance of these components. First function, cell membrane consists of lipids and proteins. That is not function, that is structure, and it can be seen through microscope. Cell membrane separates the external environment from the cytoplasm, and it gives a definite shape and size, and it also protects the cell from the external environment. And just now I told you what is that, it helps in maintaining, okay, balance. Any substance entering or leaving the cell, say definitely this is a cell, yes, no, some substances should enter the cell, some substances should leave out of the cell. Some substances should enter the cell, some substances should be leaving out of the cell. Then who will decide which substances should enter, which substances should go out? Yes or no? Which substances should enter, which substances should go out? So this will be decided by the plasma membrane. So any substances entering or leaving the cell can do only through plasma membrane. So the uniqueness of this membrane lies in the fact that it does not allow every substance. So this is a different, very special membrane, children. It won't allow all the substances, only some substances can be allowed. Okay, so the exchange of substances through the cell membranes happens selectively. That is why it is called as selectively permeable membrane. So by selectively it is happening, yes or no? Only selected substances can be entering inside. That is why the plasma membrane is called selective permeable membrane. selective, yes, selective permeable membrane. What is the meaning of permeable? The ability to pass through. So what I am telling here is, not all the substances should enter inside the cell. The plasma membrane will decide which substances should come in, which substances should go out. Yes or no? That means selectively it is allowing the substances to pass inside. That is why it is called as selective permeable membrane. Why it is called selective permeable membrane, children? Only some substances are coming inside, not all the substances. These characteristics of membranes enable us to control the exchange of substances. Yes, no. Only some substances, not all substances, means definitely this is a good characteristics, and it is controlling the exchange of substances. Some gases should come in, some gases should go out. Some liquid should come in, some liquid should go out. Some nutrients should come inside. Yes or no? Then which of them should come inside, which of them should go out will be decided by the plasma membrane. That you will learn in our chapter number 4, okay, in detail. And next one is cell wall. Next one is cell wall. Children, where does the cell wall is present? In this one or this cell? Say in plant cell only the cell wall is present. In animal cell the cell wall is absent. Cell wall is very, very, very important for the plant cell. Okay, this is a unique feature seen in the plant cell only. This is absent in animal cell. Okay, while the cell membrane acts as the outer layer in the animal cell, in plant cell the outer layer is mainly cell wall. See, this is the outer layer. Why? Because the cell wall is absent here. Yes or no? Here the cell wall is present. Here the cell wall is present. So if, sir, for example, if you will wear sweater, then if I'll ask you to remove the first cloth from your body, definitely you will remove the sweater only. Yes or no? Not your shirt. Here also the first one is only, this one only, cell membrane. But in this case the first cloth, like that first cloth or first one, protective layer is cell wall. Protective layer is cell wall, right? So while the next one, this is considered to be one of the major difference. I already told you the difference. In plant cell, cell wall is present. Cell wall is absent in animal cell. The cell wall, let us go for the features of the cell wall, is a tough, where a cell membrane is flexible, but cell wall is tough but flexible. Porous layer that tends a definite shape and to the cell protection. Okay, so the cell wall is a tough but flexible porous layer will be present that gives a definite shape, okay, and it protects the cell. Next one, earlier it was believed to be inactive, but now it is considered to be the most significant parts of the cell that continuously exchange information with other cells during growth and development. Earlier it was believed that the cell wall was inactive, nothing with the cell wall, but as the science is developed, as the progress, the science is progress, scientists still are working, yes or no? So they have came to one conclusion that earlier it was believed that cell wall, okay, was inactive, but it is very important. Now it is that they have realized that cell wall plays a key role. Okay, cell wall plays a key role in exchanging of the information. So what is the role of cell wall in plant cells? So it exerts an inward wall pressure to resist the outward wall pressure exerted by the cell sap. So this is the cell sap, which is pink in color. So there will be pressure from inside to outside. When there will be pressure from inside to outside, then there will be definitely one protective layer that is called a cell wall. Cell wall will exert the pressure from outside to inside so that both the pressures are balanced. If the cell wall is not there, then there will be an imbalance. So that is the main function. So here this is the cell wall. When there is a pressure from inwards to outwards by the cytoplasm, definitely this cell wall withstands this pressure, and this also keeps the pressure from outside to inside, so that the entire thing is balanced. Hence the plant cells can withstand much greater changes of surrounding medium than the animal cells. That is why this is the special features why the plant cells can withstand much greater changes, means because of the presence of cell wall. So once again I repeat, cell wall is the extra layer that is present in the plant cell, and this is made up of, and already I told you here, see children, cell wall is made up of cellulose. Cell wall is made up of cellulose. The cell wall is made up of cellulose. Okay, and cell membrane is made up of, I told you clearly, that is made up of lipids. That is made up of lipids, and this will be present in only plant cell and absent in animal cell. This is tough but flexible porous layer decides the shape, and it helps in protection also. Earlier it was believed that it was inactive, but here now it is very important. The scientists have realized that it is a very important, significant parts of the cell that continuously exchanges information with other cells during growth and development. During growth and development, the cell wall coordinates or exchanges information with other cells, and it exerts an inward wall pressure to resist the outward wall pressure. Outward pressure. Okay, it exerts an inward pressure to resist the outward pressure from by the cell sap. Cell sap is nothing but cytoplasm. Hence, plant cells can withstand great changes than the animal cells. Okay. In the last class, we have learnt about the differences between plant cell and animal cell. So what is the main difference between plant cell and animal cell? In plant cell, cell wall is present. Okay, in plant cell wall is present. I am writing the differences here. Differences between plant cell. Next one is animal cell. Next one is animal cell. Okay, so in plant cell, cellulose, that is cell wall. Cell wall is made up of cellulose. So cell wall is present. Cell wall is present. So in animal cell, cell wall is absent. In animal cell, cell wall is absent. This is the first major difference between plant cell and animal cell. Let us go for the second one. In plant cell, vacuoles are present. In plant cell, vacuoles are present. In animal cell, vacuoles are absent. So what are these vacuoles? What are these centrioles? We will learn in next class. Okay. So third one, in plant cell, centrioles are absent. In plant cell, centrioles are absent. And here centrioles are present. So these are the major differences between plant cell and animal cell. These are the major differences between plant cell and animal cell. In plant cell, cell wall is present. In animal cell, cell wall is absent. Here in plant cell, vacuoles are present. In animal cell, vacuoles are present. If they are present also, they are very, very small. And third one, in plant cell, centrioles are absent. See, centrioles participate in cell division, children. Okay, in animal cell, centrioles are present. Now, in the last class, we have learnt about the functions of plasma membrane. Plasma membrane is otherwise called as cell membrane. Yes or no? Plasma membrane is otherwise called as cell membrane, and this is the outermost layer of the cell. Okay, the cell membrane is the outermost layer of the cell, and that separates the cytoplasm from the external environment. So what is cytoplasm, children? Cytoplasm is nothing but the liquid or the fluid content that is present inside the cell. That is present inside the cell is called as cytoplasm. So here the cell wall, the plasma membrane separates the cytoplasm from the external environment. Okay, and the cell membrane defines the shape and size of the cell. So it defines the shape and size of the cell, encloses the cytoplasm and protects it from the external environment. The internal environment of the cell is different from that of outside. Inside a cell, one finds a very specific composition of various substances which maintains a balance. So this cell membrane helps to maintain that balance. And here any substance that enter into the cell or goes out from the cell will be decided by the plasma membrane, and this plasma membrane allows only certain substances to pass through, not all. So selectively it is allowing the substances to pass through. That is why this is called as selectively permeable membrane. So, and about cell wall we have learned, yes or no? Cell wall is the extra layer that protects the plant cell. That cell wall is absent in animal cell. We have learned this. Okay, that cell wall is very tough but flexible, porous layer that lends a definite shape to the shape and protects the cell. And earlier it was believed that it was inactive, but now it is considered to be the most important, okay, significant parts of the cell that continuously exchange information with other cells. Okay, so this we have learnt in the last class. Now let us go for each and every cell organelle that is present in the cells. First of all, we will go for the nucleus. Children, you have observed the nucleus is present in both the plant cells and animal cells, yes or no? And this nucleus is very, very important room. Nucleus is considered as cell's control room. Nucleus is called as cell's control room. Now let us learn this the first one, very, very important lab activity. So what is that, children? Nucleus. What is that? Nucleus. So observing the nucleus. So this is a lab activity. So this is lab activity. Here, see children, if you want to write any experiment, you have to follow certain things. First one, you have to write the aim. Okay. What is our aim here? To observe the nucleus in cheek cells. So cheek cells are animal cells, yes or no? In 8th class you have learned. To observe the nucleus, in cheek cells. So here these are cheeks now, yes or no? So just we have to scrape a little amount of the scrapie from the mouth, and we have to observe the scrapie by inside the electron microscope. Okay, through the electron microscope we can observe. That you have already learned in your 8th class. So now our aim is to observe the nucleus in cheek cells. So what are the materials that are required? So this is the second step. Materials are apparatus. See, if you want to write any experiment, you have to follow certain rules. So here, materials required. So what are the materials that are required here? See children, what are the materials that are required? That means what are the materials we will take in that experiment. First one, a toothpick. Okay, what is that? Toothpick. You know toothpick is very well, yes or no children? Toothpick or ice cream spoon. If toothpick is not available, you can go for ice cream spoon. A toothpick or ice cream spoon or spatula. Anyway, anything you can take. If toothpick is available, you can take toothpick, or ice cream spoon is available, you can take ice cream spoon. Or spatula. Spatula is a type like ice cream spoon only. You can. This is a laboratory one. Okay, you can go for the spatula also. So first step is to observe the nucleus in cheek cells. And here the materials required are toothpick, ice cream spoon or spatula. And next one, glass slide. What is the other one we want? We have to take Yes, glass slide. See children, this is a glass slide will be like this. Okay. So the other one is the glass slide, and the next one is here glass slide cover slip. What you have to take? Cover slip. We have to take cover slip. So this cover slip will be like this. And next one is watch glass. Next one is watch glass. All these are the laboratory apparatus, children. Watch glass. So this watch glass will be like this. Okay, first one is toothpick or ice cream spoon or spatula. Any one, not three, any one you can take. Glass slide. Next one is a glass slide, cover slip, watch glass. Next one is needle. You know needle very well, children. Yes or no? Yes. Okay, needle, a blotting paper, or you can take tissue paper also, no problem. Blotting paper. Blotting paper. Next one is 1% methylene blue. This is a 1% dye. Actually, this is a dye which gives the color. 1% methylene blue. 1% methylene blue. And next one is normal saline. Next one is normal saline. That is a glucose actually, normal saline. That is sodium chloride solution. Okay. Next one is here you can say toothpick or ice cream spoon or spatula, glass slide, cover slip, watch glass, needle, blotting paper, 1% of methylene blue, normal saline, glycerin. Next one is glycerin. This is also one, glycerin. And finally, microscope. What is the use of microscope, children? Yes, we have to observe the nucleus in cheek cells now. So with naked eye, we can't see. With naked eye, we can't see. Yes or no? So with the help of a microscope only we can see. Right? Once again, I repeat the materials that are required here. Toothpick or ice cream spoon, or spatula, glass slide, cover slip, watch glass, needle, blotting paper, 1% methylene blue dye, saline, glycerin, and a microscope. Now let's go for the third step, that is procedure. Procedure is nothing but what is the process you are going to do with all these apparatus or with this material. Let's go for that. So that is the procedure. See children, first of all, wash your mouth. Why? Because we are taking the scrappy or the thing from our mouth only, from cheeks, no? So not from outside, from inside. So that before going to take that scrappy, you have to wash your mouth. Wash your mouth and scrape a little of internal lining of your cheek inside your mouth. So here see, this is internal lining. Okay, we have to just take the toothpick or ice cream spoon or spatula. So see, the toothpick will be having two ends, no children? So like this the toothpick will be having. One is the blunt one, the other one is the, here see, this is the sharp one. Okay. With the help of the blunt one only you have to scrape, not with the sharp one. If you scrape hard, it may, your internal lining of your mouth may get hurt and it causes bleeding. That is why with the blunt end only you just scrape, not very hard. Okay, very light only you have to scrape. Okay, just little bit of scraping we want. Okay, so don't keep so much pressure there. Okay, with less effort only, okay, you can scrape it. Or if this is not available, which one, toothpick is not available, you can take ice cream spoon or at least a spatula. So before going to scrape the internal lining of your mouth, you just wash your mouth. Okay, wash your mouth and scrape a little of internal lining lining of your cheek inside your mouth with a clean, okay, with a clean toothpick or spatula or ice cream spoon. So any one you can take and you just scrape the internal lining of the mouth, means here inside the cheeks it will be present, and take that scrapie. Place the scrapie in a watch glass containing a very little quantity of normal saline. So first step, just scrape with the, okay, we have taken the scrapie with the help of toothpick, In the second step, what you have to do? In the second step, take a watch glass. So that is the watch glass is there now. Take a watch glass. See, I am taking the watch glass here. And here, inside this watch glass, here see, what you have to take? Saline. What is this, children? Saline. Inside the watch glass, you have to take saline. So here, place the scrapie in a watch glass containing a very small little quantity of saline. So this is a little quantity of saline, and anyway you have extracted, you have taken the scrapie. Place this scrapie. So this is scrapie in a watch glass containing the saline. So that is the second step. And third one, here, then place the material on a glass slide. So then place the material, this scrapie plus saline, okay. So what you have to do, here you have to place this material. So this is a scrapie plus saline, no? This is a slide, glass slide. So once again I repeat from the first. Before extract a little or take a little amount of scrapie from your internal lining of the mouth. Before going to take that, wash your mouth, okay, and with the help of a clean ice cream spoon or spatula or toothpick, you have to take the scrapie and place the scrapie in a watch glass containing the saline. And now shift this, now place the material, shift this material onto the glass slide. And next one, put a drop of methylene blue and wait for a couple of minutes. So the next step is, what you have to do? Put a drop of methylene blue. Already we have taken the methylene blue here, yes or no? So in the apparatus, so put a drop of methylene blue and wait for a couple of minutes. Where you have to put a drop of methylene blue? On the material which you have taken on the slide, no? On that you have to place this methylene blue. Okay, so here then put a drop of methylene blue and wait for a couple of minutes. Couple of minutes means 2 minutes. Right. Wipe off the extra stain with a fine cloth or blotting paper. So when we are taking on the slide, what happens? So there may be a chance of spreading here. So with the help of this blotting paper or a cloth, you just wipe off. Okay, so that the material is at the center. And next one, put a drop of glycerin over it. Again you have to, what you have to do? Put a drop of glycerin. See children, how the material is taken. First of all, we have taken the scrapie from our mouth with the help of the toothpick, and we have placed this scrapie in a watch glass containing a little amount of saline, and we have shifted this material onto the slide. Okay, and after that we have added a small drop of methylene blue to this and wait for a couple of minutes, and after that we have added glycerin. Okay, right. So place a cover slip. Now what you have to do? Place a cover slip. So what happens here? When you will place this liquid, so the liquid may be spreading and on the entire slide. So that is the reason. So this place this cover slip so that the material will be placed in the material will be present in the middle of the slide. So that place a cover slip place a cover slip, tap the cover slip with the blunt end of the needle. So we have taken the needle also here, yes or no? So what is the use of this needle? So needle will be again having the two ends, one is the blunt end and one is sharp end. So after keeping the cover slip on the center of the slide, you just tap with the blunt end of the needle like this, blunt end of the needle. Okay, so next one, tap the cover slip with the blunt end of the needle, blunt end, two ends will be there, sharp end and the blunt end of the needle, so as to spread the cells, so where the cells will be spreaded in this area only, but it won't come out from that cover slip, only in this area. So our procedure is ready. So once again I will tell the procedure. You just, before going to take the scrap from your internal lining of the mouth, wash your mouth. Okay, so with the help of a clean toothpick or ice cream spoon or spatula, take a little amount of the scrappy and place the scrappy in the watch glass containing saline. Next one, place the material on the center of the slide, and after that what happens when you place this material in the center of the slide, there may be a chance of spreading on the entire slide. So the excess stain will be cleaned with the help of the blotting paper or the cloth, and after that add methylene blue, and wait for a couple of minutes. After 2 minutes you add glycerin, and after that cover it with a cover slip and take the blunt end of the needle and tap over the cover slip, so that the cover slip will be at the center. Okay, so that the material is at the center. Alright, here our procedure is ready. Aim is to observe the nucleus in cheek cells, and the apparatus we have taken, and the procedure is ready. Now let us go for the precautions. See, there are certain precautions here. So what are the precautions that are there? See children, precautions means while doing this experiment we have to take some care. Yes or no? So first precaution, already I told you, do not scrap the cheek too hard, so that if you scrape the cheek too hard with the toothpick or ice cream spoon, what happens children? It may cause injury in the buccal cavity and causes bleeding. So that is the first precaution. Second one is scrapped material. The scrapped material should be spread uniformly on the slide. So okay, so with the help of the blunt end of the needle we are tapping now, so that makes the scrapped material should be spread uniformly. Okay, right. Next one is excess stains should be drained off. So when we have taken this scrappy plus saline here, there may be a chance of excess stain that can be drained off. Drained off means it should be removed. And next one, there should be no air bubbles under the cover slip. So please take care that there should be no air bubbles. If air bubbles will be formed, we can't see in the microscope what we want. So these are the 4 steps. First step is do not scrape the cheek too hard, as it may cause injury. Second one is scraped material should be spread uniformly on the slide, equally on the slide. Third one, excess stains should be drained. Okay, drained off. Fourth one, there should be no air bubbles. Now let's go for the observation. So what did you observe here? So observe the temporary mount under the low and high power of microscope. Using the microscope there will be resolution, low power and high power. Yes or no? Low power and high power. So here low power and high power. You can take low power and high power children. Low and high. Yes or no? So here observe the temporary mount under the low and high power of microscope. Draw your observations. So after doing this experiment, what is our main target? To observe the cheek cells under the microscope. Yes or no? So then finally you have to observe. So what you have observed you have to draw. And there are few questions here. What is the shape of the cells that you have observed? That is the first question. And second question is, are these cell structures similar to the structures in onion peel cells? Are these cell structures similar to the structures in onion peel cells? Okay, that is the second one. And the third one is, are there any dark color specific spherical or oval dot like structure near the center of the cell? So these are the three questions. So what is the shape of the cells that you have observed? And the second one is here the shape of the cell. Actually we have taken which cell, children? Animal cell. Yes or no? So this is an animal cell, okay, and this is a round circular, that is the shape. And are there any cell structures similar to the structures in onion peel cell? Yes, of course. Okay, so in onion peel cells also the nucleus will be present. We are observing the nucleus here. And the third question, are there any dark color spherical or oval dot like structure? Yes, that will be present. That is nucleolus. Nucleolus is a dark oval dot like structure near the center of the cell. So we have already studied about this dark color dot in a cell. This dark color dot, okay, so actually this is a cell. We have taken the cell. Yes or no? We have taken the cell. Which cell? Cheek cell. And inside the cheek cell, what is the shape of the cheek cells? It is almost circular in shape, whereas the onion cells are rectangular in shape. And the second question is, are there any similar structures in the plant onion cells also? Yes, here the nucleus is present, there also the nucleus is present. And inside the cell, yes, there is a oval dot like structure you can observe here. That is what's so called as nucleus here. Our main target is that one only. Okay, so inside the nucleus again nucleolus is present. That is the second matter. Okay, so there also in plant cells also the nucleus is present. So nucleus is present in plant cell, nucleus is present in cheek cells, animal cells. That is the common. And the difference is here the cells are circular, and the cell is circular in shape, there the cell is rectangular. Onion cells are rectangular in shape. That we have learned. Okay, so this is about the experiment. So we have seen the aim, apparatus required, the materials, precautions and observation. Okay.